So let's talk about DNA sequencing. So what DNA sequencing means is that DNA sequencing technology is a way to figure out the exact arrangement of nucleotides inside a DNA molecule, which is absolutely super important for understanding into what protein that DNA sequence can be translated. And the traditional approach that's still used for your standard sequencing reactions is the dideoxy-DNA sequencing. It's also called Sanger sequencing, and it uses nucleotide chain terminators. Now, here's the picture of a typical normal deoxynucleotide used in a PCR reaction that is easily incorporated into a growing DNA strand by the DNA polymerase because it has a 3' OH group. Here the molecules flipped. During other talks, I had the phosphate groups on this side, but this is the 3' OH, and this is the 5', which holds the triphosphate. Now, the chain terminators have been modified. They lack the 3' OH group. As a result, DNA polymerase cannot catalyze the reaction. It needs to deproteinate the OH group in order to catalyze the formation of the phosphodiester bond with the next nucleotide. So while these guys can be incorporated into a DNA molecule, no new nucleotide can be added. Because as the nucleotides come in, the bond is synthesized 5' to 3'. So if you pretend there's a DNA molecule here, a 5' phosphodiester bond is present. But the next bond cannot be catalyzed because there's no OH group in that location. There's just one hydrogen. As a result, this is how the reaction works. You have a Taq DNA polymerase extending the strand, and here the OH was able to be deprotonated, and as a result, the pyrophosphate has been released, and a phosphodiester bond will be catalyzed. While this molecule, the dideoxy, both OH groups missing, the dideoxy ATP will be incorporated into the nucleotide, the growing DNA chain, but will not allow incorporation for any more nucleotides. It's a terminator. It will terminate the DNA polymerase reaction. Now, this can be used because you can make these dideoxy ATPs, TTPs, and so forth, and you can attach fluorescent molecules to them. Usually they're attached right next to the base pair. And they come actually in 4 different colors. And so when Sanger sequencing reaction is set up, you have 1 primer instead of a PCR reaction where you have 2. So the deoxy sequencing reaction uses 1 primer and a template of unknown sequence. And then a DNA polymerase tag or another DNA polymerase is added, as well as 4 different kinds of dNTPs and high concentrations, plus 4 different kinds of dNTPs, the dideoxy NTPs that will terminate the reaction in lower concentration, because you don't want them to incorporate right away. You just want a few of them, and you want the polymerase to be primarily able to extend the chain. So when the DNA polymerase and the nucleotides have been incorporated, the polymerase has extended. At some point, it grabbed the dideoxynucleotide instead of the standard one, and that chain was terminated because DNA polymerase could no longer synthesize the reaction because the OH group was missing. And then you can you allow this reaction to proceed for a while to assure that once you have 25 cycles, the thousands of template molecules have been created, and there's enough of each molecule for every nucleotide. So there's a DNA molecule with a termination nucleotide incorporated at each position of the sequence has been created. So you guys can see that here. Now, here, at this point, it's still a mixture of lots of different nucleotides, all fluorescently labeled at the very end, with the terminator. nucleotide. They're all mixed together inside a PCR reaction tube. The next step is to separate them by size. And the separation is done is done using the gel electrophoresis principle. So all of these DNA molecules are put into an electric field, and they will all migrate at different speeds towards the plus end. And they all have to force themselves through the gel-like medium. And the shorter ones will do it faster, and the longer ones will do it slower. But instead of using a gel electrophoresis, because the typical agarose gel, which their holes are much too big to separate different DNA molecules based on size, instead a capillary gel is used that allows for separation of single nucleotide of DNA molecules of different single nucleotide length. And as these DNA molecules travel along the gel capillary, they pass a laser beam that will that wavelengths will activate all 4 of the fluorescent molecules, but they will emit in different colors, and the different colors of the emission will be detected. So based on fluorescence, if it was green, they know this was the final terminator. Here was an A, because only A could have base paired with whatever it was making. And then it was a T, then it was a C, then it was a G, etc. And then you get this fluorescent readout that first, the smallest molecule that passed by the laser beam glowed blue, so it was an A. Then the next molecule also glowed blue, so it was also an A. Then a T, then a C, then a G, then a T again, and so forth. And you can see that because the laser beam is actually detecting actual fluorescence, there's going to be some sort of leakage of other fluorescent signals if certain mistakes were made by the DNA polymerase, or there's a certain detection error. But as long as you have a nice big peak for each of your nucleotides, you know that your DNA sequencing reaction was efficient. It worked well, and when you see double peaks or 2 colors coming up, you will know that there is an inconsistency in that area. Either some of your template had a mutation in that area, or the DNA polymerization reaction was inefficient there due to like a long stretch of T's, for example. But so this is the basic principle of DNA sequencing using the Sanger method. You run a PCR, you use one primer, you use a PCR-based amplification using one primer, and you use dideoxy terminators that will be incorporated by the DNA polymerase at different places of the DNA template of the DNA molecule. Then you take these DNA molecules and you separate them by size, and you detect based on the fluorescence of your dideoxy terminator for each of these DNA molecules, whether or not what kind of DNA nucleotide was incorporated at that very last step. And this method and the machines that perform DNA sequencing have been so well optimized by now that you can get about 1,000 base pairs from a single run. It didn't used to be this efficient. In the past, people actually published papers. This was enough for publication that they sequenced a stretch of DNA that was about 200 nucleotides long. So here's another lecture question that hopefully you guys will try to attempt to answer before I give away the answer. So the idea here is to figure out which of these primers, A, B, C, or D, will be the most useful for sequencing the entire gene. In this case, it's going to be gene for red. Now, the answer is you can use B. B will read in this direction and will give you the sequence for the entire gene, which if the gene is 600 base pairs long, you will have no problem getting the entire sequence from the sequencing facility. It binds at the beginning of the gene and it will give you the sequence for the whole thing. Now, you can also use D. The D will give you the sequence in the antisense orientation, so it will be reverse and complementary to the 600 base pair long gene, but you can reverse and complement the sequence and get the correct stuff. C will only provide the sequence for the 3' region of after the gene, and A will only provide sequence in this direction of the 5' region of the before the gene, not of the actual gene, so maybe where the promoter is lying.